Hello, ma'am. Good evening, ma'am.
Good evening, sir.
Good evening, ma'am.
Good evening, sir.
Good evening, Varsa. Good evening.
Good evening, sir.
Good evening, Ajay.
Sir, you can start now, sir.
Yes, ma'am. Thank you. Thank you so much, ma'am. Okay. Well, so good evening, one and of all my dear students. Last class, what we have discussed in our about polygenic inheritance. This was introduced by Nielsen and Egil. He is only one scientist, sir. Okay. And then next one is the sex determination, how it will be determined by the scientists. What are the examples? Okay. What is by X chromosome? What is by Y chromosome? Who discover? How many genes are there? How many genes are identified? That thing's also already we discussed, isn't it? X chromosome was discovered by the name of the scientist Hermann Henking. Y chromosome already you know about Stevens. Yeah, okay, the Y chromosomes. But number of genes, 1,100 genes are present. Okay, from these, how many genes are identified? Known one, 231 only. Okay, that things also. And what are the type of sex determination on the basis of fertilizations? Okay, there are 3 types have been reported. One is called as progamic, and second one is syngamic and epigamic. Don't forget, my dear students. Progamic before, syngamic at the time of fertilizations, and epigamic that after fertilization. Already you know what are the examples. Okay, that things already we discussed. And what is by autosome? Types of chromosome. There are 2 types of chromosome according to their properties, isn't it? There are 2 things: the autosome and allosomes. How they are, that things also already discussed. And these things also. Okay, how it will be given or what is the nominated, like that we may tell us. XX, XY type, it's also called as Legness type. Another one is called as, what is that? X in reversible, reciprocal manner. Okay, that things also already we discussed. And then that is called as which plant we were selected. They were selected, strawberries. That things also already we discussed, isn't it? Okay, so next determination in human beings already you know about. There are 2 types is that. One is called as heterozygous condition and homozygous. Mostly female or homozygous XX already we discussed. And father is a heterozygous because they produce 2 types of gametes, X and Y. So corresponding fusion, as a result, these are the, what is that, combination we will obtain. Okay. Okay. So this is also a same one. And this was first sex determination in plant also identified. The name of the scientist called as Warmack, and the plant name is called as Melandrium album. And then which type of chromosome present in it, and that things also. Not only that, and also another plants also. Name of the scientist R.P. Roy gave the importance of Y chromosome in plant. Okay, discovered in sex determination in Coccinea indica, family is Cucurbitaceae. Okay, sometimes they may ask the question, Coccinea indica is also comes under which type of family, which family, like that. Okay, so this is also, and how many regions also there. Y chromosomes, it is a long one, so it contains, what is that, 4 region is there. X chromosome is a short one, that's why it is contain 2 regions. Totally 6 region is there. Each one it has a specific properties. Okay, so these are the things already we discussed. What are the responsibility? That things also already discussed. What is one? First one is female suppressor, male promoter region. That things also already we discussed in the previous classes, isn't it? Okay, so today, and genetic disorder also we discuss in slightly. Okay, what is my genetic disorders? Genetical changes occurs due to the addition, deletion, substitution, inversion, and also there are so many things is there. Okay, a particular nucleotide is removed from the particular gene is removed from the what is that nucleotide. What happened? It may change tremendous changes which occurs in the nucleotide. As a result, the protein molecules also, and also some diseases also produced that was identified in pedigree analysis. It is also one of the identification, historical aspects, ancestral. Okay, that things also already we discussed. Okay, so these are the genetic disorders. They are using some symbols already we discussed, male, female, mating, and so and so. Okay, and so this is also already we saw it in the previous classes. Okay. Okay, pedigree analysis provide valuable information regarding genetical makeup of human beings. If any genetic disease is occurring in a family, already I told you, some of the persons, our father, maybe grandfather also suffered by or affected by the sugar or diabetes or something else, whatever it may, color blindness and whatever it may be, that can be identified. Suppose we do not know whether the grandfather, grandmother having a presence or not, like that, we do not know, isn't it? That things we can easily identify through this pedigree analysis. Okay, Then the pedigree analysis provide guidance to forthcoming parent about their future progenies. For example, polytractile allele in humans. This is one of the, what is the example has given, already we discussed. This is also one of the genetical disorder. How can you identify it? By the pedigree analysis. Okay, see that. Okay, this is also already discussed. Okay, humans will have, now we are going to discuss about the how it will be, what are the types of chromosomes, how they are classified. Okay, that things also, what are the two types of disorders there. One is called as Mendelian disorder, another one is called as chromosomal aspect, gene aspects. That things also already we discussed in a chart, isn't it? Okay. Human have 23 pairs, otherwise called as 46 chromosomes. Yes, correct. In this method, the chromosomes, okay, the chromosomes mean total. So what are the chromosomes? Two types is. One is autosomes, another one is called as sex chromosomes. Are arranged according to their size and structure. Don't forget, my dear. They may ask the questions according to their functions or structures or size, like that. They may ask the questions. They are arranged in their according to their size and structure. Note it down. Based on the position of the centromere. Don't forget what is meant by centromere. You know about each and every chromosome. They are having one. What is that? Hands like that. There are two hands there. One is called subunit. They may call it as a subunit. One is called as large arms, otherwise called as short arms, like that, isn't it? They are united with the or connected with the central point is called as centromere. Okay, see that one. The both on position of the centromere and the relative length of the both arms of chromosome, they are classified into 4 types. Okay, not only the 4, 5 is there. Okay, of chromosome are found in human beings. What are the types? Here 46 chromosome is there. This 46 chromosome is also present in, they may exist in 4 types only, depends upon their, what is the position of the centromere and length of the arms. Okay, this is also very, very important. According to this one, they may be divided into 4 types: metacentric, submetacentric, telocentric, and acrocentric. Okay, see that structure, you can easily identify. First one is called as telocentric. Where is that centromere? Here the centromere is there. Okay, this one is present or absent. This is also present in your only. The only one arms is that present in it. With long arm only attached with the centromere. This type of chromosome is called as telocentric. Acrocentric. Acrocentric is, there are 2 types of centromere. 2 types of arms also connected with the centromere. One is a long one, another one is a short one. This one is a long one, and the upper portion is a short one. Okay, connected in the straight line. Okay. Submetacentric. Submetacentric is also divided. See, having 2 arms is, one is short, another one is also a long one. It's look like a L shape. That is very, very important. Straight structures is called as, what is it? Acrocentric, straight nature. Just like a draw a 1. Okay. So center there is a, what is a centromere is that the connection of the 2 arms, the long one as well as a short one, connected with the help of the portion or point is called a centromere. Okay, and then submetacentric means the arms are short and longs also connected with the center portion is called a submetacentric. It's look like a L shape. Another one is called as metacentric. Both arms are what is that? Equal in portion, equal portion connected with the central portion is called as, what is that? Centromere. Such type of chromosome are called as, what is that? Metacentric. These are the 4 types of chromosomes present in human being. Okay, so these are the, how they are classified means depends upon their position of the centromere and of the arms. 2 arms, one is short, one is long. Okay, any doubt in this area, Mike?
No, sir.
Okay, good. So go to the next one. The karyotypes help to know the relative structure, morphology. Karyotype means, you know, it will carry on. That is nothing but the nucleus. Okay. We can easily identify the structure of morphology of the chromosomes. Besides, it helps in the chromosomal identification and its nomenclature. It is also used to studying the chromosomal abnormalities. Why we are studying the morphological structure of the chromosomes? Okay, you know what about the The chromosome is made up of what is that? Gene. Gene also present alleles. Alleles present in the a particular portion is called as locus, isn't it? So suppose any one of the gene, take an example, a particular gene, A is an example, is responsible for the color blindness. C, you can imagine, C, color blindness, like that. C is responsible for the color blindness. Okay. And then another one is diabetes. D is denoted as diabetes mellitus, like that, we can identify which chromosome, which portion, which alleles responsible for the, which one, the particular disease is causing. Okay. So we can identify it. So that is the things. Okay. The morphological structure mostly used for the identification and also study of the chromosomal abnormalities. And here the positive aspect is also there and negative aspect is also there. Okay. See here. So now already we discussed in the chart, already we saw it. There are two types of disorder is there. One is called as Mendelian disorder. Another one is called as chromosomal disorder. Here we are going to discuss an expansively. Okay. Mendelian disorder. Already broadly genetic disorder may be grouped into two categories: Mendelian disorders and chromosomal disorders. Okay. What is my Mendelian disorder? See here, the Mendelian disorders are mainly determined by alternation or mutation in the single gene. Note it down here. Here the gene only responsible for the particular diseases. Here total chromosome. Okay. So okay, this is good. This disorder are transmitted to the offspring on the same lines as we have studied in the premise of inheritance. So this is also a important one. Okay, principles. The pattern of inheritance of such Mendelian disorders can be traced in a family by the pedigree analysis. Suppose any one of the parent or any one of the grandparent suffered any one of the disease by a particular gene or recessive gene from the X chromosome, or whether it may be a Y chromosome, whether dominant alleles, whether it may be a recessive alleles. How can you identify? Means with the help of Mendelian disorder can be analyzed, traced out by the pedigree analysis. Most common and prevalent Mendelian disorders are normally they may be identified. What are the things? Hemophilia and cystic fibrosis and sickle cell anemia and color blindness and phenylketonuria and also thalassemia and also etc. These are the, what is that? Common prevalent Mendelian disorders. Mendelian disorders, particular gene is responsible for hemophilia, particular gene, recessive gene is responsible for the cystic fibrosis and sickle cell anemia and color blindness, phenylketonuria and also thalassemia respectively. Each and every genes, okay, a particular gene responsible for the particular disease. How can you identify which gene is responsible for the hemophilia? That time, what happened? The chromosomal, okay, chromosomal structure, nomenclature. You have to go through it. Then only you will get the exact gene is responsible for the diseases. Okay. Okay. What are the diseases? Here we are going to discuss. See one by one. Once again, repeat for you. Okay. See this one. Mendelian disorders. The Mendelian disorder is also, what is that? Nothing but divided into two categories: Mendelian disorder and chromosomal disorders. The Mendelian disorders are mainly determined by alteration or mutation. What is by alteration? Suppose the adenine present in it, suppose in the position of 15th, just replace it, add another nucleotide. Okay. And mutation is also a similar. Okay. Or mutation means what is that? The sudden changes takes place due to the physical, chemical, and also biological process. In particular, gene will be completely removed from the particular, what is that? Nucleotide or chromosomes or whatever it may be, particular gene. Okay. It's also impact the next generation. Okay. It will be produced there. Okay. That things. Okay. See here. So what are the diseases? How can you identify? With the help of the pedigree analysis only. What are the things is that? These are the things is here. Okay. Hemophilia, cystic fibrosis, sickle cell anemia. What are the gene responsible? How it will be? Okay. Last class already we discussed. What is by neurospora? Neurospora is nothing but one of the fungi. Okay. In genetic biotechnology and genetic engineering, they were used in the research purpose. Okay, see here. So Mendelian disorder may be dominant or recessive. Already I told you. It is depends upon the dominant gene or recessive gene. Okay. By pedigree analysis, one can easily understand whether the triad in question is dominant or recessive. Already told you. Okay. Dominant or recessive. Okay. Positive or negative. Whether it may be X chromosome or Y chromosome. That is very, very important one here. Dominant, whether X chromosome or Y chromosome. Recessive, whether X or Y, like that. Okay. See here. The triad may also be linked to the sex chromosome. As in the case of hemophilia, it is evident that this X-linked recessive triads, note it down, my dear student. Hemophilia is also one of the sex-linked chromosomes, but it is also X-linked recessive triads, shows the transmission from carrier female to male progeny. Okay, because the hemophilia is also one of the diseases caused by the X chromosome. X chromosome means, what is that meaning? It is a sex-linked, XX, XY, like that, isn't it? That's why the X-linked recessive gene or triads shows the transmitter from the carrier female to male progeny. Suppose the female is affected by the hemophilia. Already we know about the Victoria Maharani, isn't it? Victoria Prince, sorry. Okay. See that. That is the thing. See here, how it will be. First one is hemophilia. The hemophilia is one of the genetical disorder, Mendelian disorder, or gene disorder. Don't forget, my dear. The sex-linked recessive disease, which shows its transmission from the unaffected carrier female, to some of the male progeny, has been widely studied. How they studied, you know, unaffected carrier female to male progeny has been widely studied. This was first introduced by the name of the scientist called as John Otto. Otto Sert, you just note it on, my dear. Otto Sert. Okay, John Otto in 1803 first discovered which one, you know? Hemophilia disease. Okay, how? It is in this disease a single protein that is a part of cascade of protein involved in the clotting of blood is affected. A particular protein molecule is absent. What happened, you know? It will be completely lack of the blood clotting mechanisms. Already we studied how many factors responsible for the blood clotting mechanism, idea. Dhanalakshmi, how many factors is essential for blood clotting mechanisms? Ajay, how many factors, ba?
So, so
What?
I'm not sure, sir. Is it two, sir? Two factors.
No. There are 14 factors is essential for the
14,
blood.
sir. Yes, sir.
Yeah. Blood clotting mechanism. I think okay, try. Okay. Okay. From this one, what are the three factors responsible for the hemophilia? Varsa, do you know the do you know the answer for this one? Hemophilia. Which factor is responsible? There are 14 factors responsible for the blood clotting mechanism, isn't it?
Yes.
Okay. What are the three factors? The first factor is eighth one.
A gene,
Okay, good.
HGB gene.
No. It's not correct. I'm asking the factors. I'm not in asking the factor. Blood clotting factor I'm asking. Okay, anyhow, I will explain. No need to worry. Already, you know, there are 14 factors is the 1 to 14. Okay, from this, among these one, there are three factors responsible for the hemophilia. The 8th factor. Okay, and then 9th factor, 11th factor. Note it down, my dear students. Three factors very, very essential for blood clotting.
8th, 9th and 11th, sir.
Yeah, yes. Okay, good. So in this disease, a single protein, that protein molecule is nothing but the factors. Okay, how many factors? You know, three factors is essential for the blood clotting mechanism. Suppose if it is not like that, you know, it will affect the blood clotting mechanism. As a result, what happened? You know, completely bleeding. Suppose if it is present, what happened? The bleeding is completely stopped. Suppose if it is not present, what happened? The bleeding is leading to ooze out. Due to this, in an affected individual, affected means the lack of genes. Okay, individual, a simple cut will made non-stop bleeding. That is the reason. Already we discussed. I think you forget it. Okay, see here. The heterozygous female carrier for the hemophilia. Heterozygous means what is that? X and Y. Hemophilia may transmit the disease to sons. Okay. The possibility of the female become a hemophilic is extremely rare because mothers of such a female has to be at least a carrier and the father should be a hemophilic. Unviable in the later stage of the life. When you are going to pedigree analysis, what happened? These are the stages we can easily identify. How will you? Which one is? Heterozygous female carries the hemophilia and transmitted disease to the sons. Okay, when it's a hemophilia divisions is there. Heterozygous means XY. Okay, Y is denoted as which one? XX. Sorry, XX is there, but also affected one, carrier. When it's split into two, you will get X and also X. When it joined with the male gametes, what happened? XY is there. So that son is affected by which one? Hemophilia. Who is the carrier now here? The female is the carrier. Okay, see here. The family pedigree of Queen Victoria shows a number of hemophilic descendants, as she was a carrier of the, what is that, disease. Okay, see here how it will be possible. So this is the pedigree chart of hemophilia in royal family. Okay, Victoria Prince. So the Albert is the husband of which one? This is the symbol. But the symbol is square is a symbol of male. The round already but the Victoria. Both are married. That is called as the connection is called as what is the marriage. As a result, you will get it the possibilities. What are they? Edward, Alice, and Leopold. Okay, and also again Beta Rice. These are the what is that? Second F1 generations. Which one is affected? The male is affected here. Okay, so that then. So here, as it is, it's coming as it is. What is that? There is no, it is a normal one. What about here? They also marriage with the another. What happened? How it will be like that? See here, female and male is affected, and also female is not affected because they are all carriers. Male only affected. So then again, what happened? The males also comes here fully affected persons. See the George VI, Walter Mayer, and also Heinrich, Alexis, Alfonso, and also Gonizola. These are the boys affected by, completely affected. Okay, when you are putting the pedigree analysis, you will get the possibilities. Okay, go to the next one. See that X chromosome inheritance. The mother who is heterozygous, already we discussed carrier, isn't it? Heterozygous means the partially, but there is the characters. Partially darkened and partially is a normal one. Parents and father without hemophilia. This is a normal person. Both are married. What will be the object here? See that the son with the hemophilia affected completely 25% chances there. And heterozygous condition is partially is also the daughter. Again daughter. Again son. Two son, two daughter is no. See here, one is son is without hemophilia, just like a father, and opposite manner the son with the hemophilia. In center there is a, what is that? Daughter who is a heterozygous, a carrier. Daughter who is not heterozygous is not a carrier. Okay, you have to put here what is that? X and X and X and Y. Split the gametes. XX, what happened? XY, what happened? Okay, just like a monohybrid class, you will get in the percentage. This is the possible childs. Okay, so all are 25%, all are 25, 25, 25, 25 like that. See that this is the parental similar one, but these two, this is also a parent, just like a mother. This is one daughter is completely what is that? Not affected, but at the same time heterozygous condition is there. But what is the meaning of heterozygous? Both the character present in it. Okay, here the X is there. Here also only X is there. But this is, what is that? heterozygous, but not affected. But in this category, what is that? Affected one. Okay. This one's completely affected. Okay. This is the possibility. Okay. Next, the coagulation cascade. Already we discussed. There are 14 factors is there. Just note it down, my dear students. Starting from the fibrinogen and finishing end with the, which one, you know, active forms. Okay. See here, 13 factor. These are the another one. Okay. So here the eighth one. See the anti-hemophilic factors. Note it down, my dear students. This is the fact blood clotting mechanism. See here, intrinsic pathway surface content. Here, the started from the 12th to 12th A. So what is the meaning of A? You know, it is an active process. It is also active. So this one is also inactive. When it inactive form of any factors converted into active factor. That's why they put here A. See, these are all are A like that. Okay. So these are the factors. What are the factors involving? 14 factors is involving in this process. Okay, traditional equation is there. Traditional equation already, you know, fibrinogen converted into fibrin. Fibrin is nothing but the clad that has given here. Okay. Before that one, prothrombin converted to thrombin. In this category, prothrombin converted to thrombin with the help of the factor is called as which one, in a 10th factor. 10th factor, what is that? Stratrate power factors involving this factor. Each one is interconnected. Okay, see here. When it first one, just take it, my dear students. This one, first one. Here the 12th factor is there. Which one is the 12th factor? 12th factor is called as, what is that? Hagman factor. The 12th factor. It is an inactive form. Hagman factor is an inactive form. The inactive form converted into active form by the surface, intrinsic component. When it inactive form is converted to active form, it's also further involved to the next reactions. That is very, very important. When it converted to inactive to active, it will also lead to the another reactions. Here, 11th one. 11th inactive is also converted to 11th active by the presence of 12th factor, active factor. Likewise, interconnection, and finally we'll get it the fibrin clad also. This is the process. Okay. See that these are the, what is the factors? Fibrinogen, prothrombin, thromboplastin, calcium ion is also 4th factor. Note it down, my dear student. One time they were asked the question, the calcium ion, which number in the blood clotting factors? 4th number. Christmas factor, don't forget, my dear students. This one, the 9th factor, anti-hemophilic factor, otherwise called as B factor, AHF B. So Christmas factor, this is also very, very important. Okay. And then the 10th one, Starrett power factors. 11th one, AHF plasma, thromboplastin, antecedent. And 12th factor, Hagman factor. And 13th factor, FSF, fibrin stabilizing factors. Okay, these are all are identified in where? From the serum. Okay, and plasma. So you will get the clotting factors. When you are getting the clotting factors, you know, so serum, it will not have any clotting factor, because serum, what happened, you know, after clotting only, we will get it the yellow color fluid. That fluid is called as, what is that? Serum. Plasma means when we are adding the anticoagulant substances, that time what happened? You will get the fluids. That is called as plasma. It contains clotting factor. Okay. Don't forget, my dear. Serum, the calcium is completely absent because the calcium is utilized for the blood clotting mechanism. It is also very, very important one. Okay. Plasma, the calcium is present in it. How it will be possible? Tomorrow you have the homework. What is my serum? What is my plasma? How the calcium present in it? Serum is completely absence of calcium. How? Why? Plasma, the calcium is present, but the clotting is not possible when you are adding the anticoagulant substance. What will be the anticoagulant substances in blood? So that is the 2 questions you have to answer it tomorrow. Okay, this is your homework tomorrow. Okay, see the next one. This is the surface content. See the fibrin clad. Clotting cascade, 12th A, and 11th A, 9th A, 8th factor is also 10 A. So these are all are converted to inactive factor, converted to active factors. So after the activate component, converted to activate component, they may enter into the next reaction. They carry out the next reactions. Okay. So this is the last one, fibrin clot. Okay. See that. Okay, here the hemophilia also further divided into, according to their nature, they may be divided into 3 types: hemophilia A, hemophilia B, hemophilia C. Okay, suppose the hemophilia A, what is the meaning of hemophilia A? When the lack of, which one in the factor 8th one, otherwise called as anti-hemophilic globulins. Okay, the words are told, I think. Okay, the anti-hemophilic globulins lack. That means, what is the meaning? The particular gene, they will not produce the 8th factor. Okay, that condition was called as, what is that? Hemophilia A. Which one? Anti-hemophilic globulin, AHG. That factor is called as the 8th factor. That 8th factor is lacked. That is called as hemophilia A. Another one is hemophilia B, or Christmas factor, due to the lack of factor 9th one. Already told you, plasma thromboplastin component. Plasma thromboplastin component. Okay, the particular gene, they will not produce the, which one, the Christmas factor. The plasma thromboplastin component means that condition is called as, that gene is called as, that type of disease is called as hemophilia B. Hemophilia C means, what is it? Due to the lack of factor 11th one, plasma thromboplastin antecedent. Don't forget, my dear student, these are the very, very important. So many times they were asked the question. Particularly the Christmas factor, hemoglobin B. They may ask the question, hemophilia, how many types is there? Hemophilia A and B and C also there. And which one is responsible? Match the following, they may ask the question. Okay, don't forget, my dear. Hemophilia A, 8th factor. Hemophilia B, 9th factor. Hemophilia C, 11th factor. These two easily identified because the X is the, you have to put one before, that is 9th factor. And after that, 11th one. You have to add the 8th, 9th, 11th. 10 only absent here. Okay. So this is very, very important, my dear student. Don't forget. Okay. And next one is called a cystic fibrosis. It is also another type of hereditary metabolic disorder of the disease. Okay. Here, what happened, see here one by one. Cystic fibrosis is also another type. It is also a hereditary metabolic disorder that it is controlled by a single autosome. Autosome means, what is that there? Somatic cell, particularly recessive gene. Don't forget, it is not come to the sex-linked chromosome. It is comes under the autosomal recessive gene. Okay, so cystic fibrosis. The gene is specific an enzyme that produce a unique glycoprotein, because all the enzymes are protein, isn't it? All the proteins are not enzyme. Don't forget, my dear. All the enzymes are proteins, but all the proteins are not enzyme. This is the proverb according to the enzyme activities. Okay, here what happened? Cystic fibrosis, the particular gene is responsible for the synthesis of enzyme, the enzyme, and also the unique glycoprotein. Glycoprotein is also complex protein. This glycoprotein result in the production of mucus. Okay, listen, the particular gene produces an enzyme, and also the unique glycoprotein also synthesized. The glycoprotein is, what is that? Synthesized but by a particular gene, and also they induce the production of mucus. So what happens? Suppose the enzyme, glycoprotein, and the mucus secretion takes place. What will happen inside the body? Which is responsible? See here, the more mucus interfere with the normal functioning of the several exocrine glands. Suppose the enzyme production takes place by the single autosomal recessive gene. The enzyme glycoprotein production takes place. The glycoprotein mucus secretion takes place. As a result, what happens? The mucus interfere with the normal functioning of the several exocrine glands, including those in the skin, lungs, liver, and pancreas. These are all, okay, these are the, what is that? Exocrine glands, they told. Some of the endocrine glands also affected. Okay. See what happened. So this is the lungs, isn't it? This is the healthy bronchitis, the open airway, the good airflow. It can be easily flow in this way. Due to the glycoprotein deposition, glycoprotein synthesis, as a result, what happened? You know, the deposition of the mucous membrane on the wall of the, which one you know? Healthy branches. As a result, what happened? The diameter will be reduced. Here that there is, see the diameter, it will be reduced compared to this one. Here the large one, the width is very large in nature. Here the diameter will be reduced due to the deposition of the mucous membrane. As a result, what happened? The airflow will be very less in nature. Okay, it occurs in the lungs. What happened? The what is that? Breathing problem is also created. Okay, that condition is called as cystic fibrosis. Note it down, my dear student. This is also one of the autosomal recessive gene. The recessive gene produces a particular enzyme, and the enzyme is also responsible for the production of the glycoprotein. Suppose overproduction of the glycoprotein, what happens? You know, it is also responsible for the production of the mucous membrane. The mucous membrane deposited on the wall of the branches, healthy branches. What happened? The diameter will be reduced. So the airflow will be suffered. Okay, it occurs in the in the lungs. There is a small square is there. This is called it's affected area. We cannot be able to magnify this area. Okay, see the next one. So this is the structure. Cystic fibrosis. Cystic fibrosis target gene. This is the particular gene responsible. The particular gene is called as chromosome 7. Note it down, my dear student. Which chromosome, which number? Chromosome 7. Thick, sticky mucus, black, pancreatic, and bile duct also. Sometimes, suppose the overproduction of the glycoprotein, what happen? You know, it may be affect the pancreatic and bile duct also. As a result, what happens? The production is, it will not reach the particular, it will not reach the target cell. It is also very, very important. The above the thicky, sticky mucus block airway. Suppose in the portion of the branches, the airway blockage takes place. What happens? You know, the flow is also suffered. Which gene? Which chromosome? 7th chromosome. Don't forget, my dear students. Okay, 7th chromosome responsible for the cystic fibrosis. Okay. The cis, the third letter is cis, S. So 7 is also cystic. Likewise, imagine. So see that structure will look like this one. When you are going to take a X-ray or whatever may be, when the person suffered by the cystic fibrosis, his lungs look like this type. Okay. So this is also a important things. And third one is called as sickle cell anemia. Already we studied that one is also. The sickle cell anemia is also another type. This is an autosome linked recessive triad. So this is also one of the autosome linked recessive triads. Note it down. Take a notes, please. That can be transmitted from parent to the offspring when both the parent or carrier for the gene or heterozygous conditions. Okay. So it is also one of the autosome linked recessive triads. It can be transferred from the parent to the offspring. Already you know about parent only the, what is that? Transfer the character from the one generation to another generation. So as a result, what happened now? When both the parenter or carrier for the genes, that condition is heterozygous nature. This disease or the disease is controlled by a single pair of alleles. Note it down, my dear students. This sickle cell anemia is controlled by a how many pair of alleles? They may ask the question. This is the single pair of alleles, that is HBA, HBA and HBS. This is the single pair. This is the one alleles. This one is also another one allele. HBA, HBS. Don't forget, my dear. Out of these 3 possible genotype, only homozygous individual for HBS. HBS. So the disease phenotype. Okay, so these are the things. So once again, repeat for you. Sickle cell anemia, autosome linked recessive. They transfer from the parent to offspring. So they controlled by a single pair of alleles. What is that? You know, HPA and HPS. There are 3 possibilities there. 3 possibilities of genotype. That is homozygous individual for HPS. And second one is heterozygous nature, means what is that? HPA, HPS. Here, this is the one type. This is the another type. This one is also another one. HPS, it may be existing HPS. This is homozygous nature. See that? Homozygous. This one is heterozygous. That means what does that mean? HPA is also present. HPS is also present. That condition is called as heterozygous. Okay. What happened here? Think it. Suppose if it is heterozygous individuals appears apparently unaffected, but they are carrier of these disease, as there is 50% probability of transmission of the mutant gene to the progeny, thus exhibiting sickle cell triads. When it heterozygous condition, what happened? Automatically they may be united with the corresponding. So they are carrier, isn't it? 50% of which gene is responsible here? HPA is responsible here. See, this is the heterozygous condition, isn't it? It is 100%. So how many percentage of HPA present in 50%? Okay. This is also very, very important. See that. Here, they may ask the question, sickle cell anemia, the alleles, which one of the following is homozygous conditions? HPS, HPS. Suppose if it is heterozygous condition, HPA, HPS. HPS, HPS, homozygous, both are same. Here, HPA, HPS. This is the heterozygous condition. Here, how many percentage is possible during the heterozygous condition, sickle cell anemia? How many percentage is possible? Mean 50% is possible. Okay. So don't forget, my dear. This is called a sickle cell anemia. This is a normal one. This is a normal erythrocyte red blood cell. Double disc saved. 120 days is also there. They carry the 4 hemoglobin molecules. Sorry, 1 hemoglobin molecules carry the 4 oxygen molecules. That thing's also already know about, isn't it? Suppose if it is changed into the disc shaped structure into sickle shaped, this is the abnormal shape. Red blood cell, what happened? They will not carry the hemoglobin molecules. They will not carry the 4 hemoglobin molecules. As a result, the particular cells, they will, what will happen? Lack of oxygen molecule. They will not supplement the oxygen molecules. What happened? The metabolic process completely lack. Okay, you got it, that one. See that sickle cell anemia like that. This is the normal one. This is the sickle cell. How it will be carry the oxygen molecules? It will not carry the oxygen molecules. As a result, what happened? The problem is created by the person. Okay, this is due to the what happened? It is also due to the number of which chromosome number. Note it on. See that this is also very, very important. See here. Here, what happened? This is the gene GTC and GAG. These are all nucleotides. The mRNA already in our board. This is a GAG. So what are the molecules? What are the protein molecules present in it here? HPAs here. How many nucleotides? How many amino acids present in it? Valine, histidine, leucine, threonine, proline, glutamic acid, up to 1 to 7 only. Okay, which one? HPA peptide. This is the A peptide component, the protein molecules. Okay, see that one. HPA peptide chain. So these are 1, 2, 3, 4, 5, 6, and 7. 7 amino acids also connected with the which one? Peptide bond. Already know it. Protein molecules are connected with the peptide. That's why they have given as a peptide here. Protein also linked with the peptide bond. Okay, already know about. Glucose with the glucose, which linkage is there? Please, anybody else, my dear? Glucose connected with the another glucose molecules. That bond is called as, what is that? Yes.
So covalent bond.
No, where? Sorry, anything else? Covalent bond is there. You have a specific term is there. You have a specific term. What about Ajay?
So glycosidic bond, sir.
Very good. Glucose molecules are connected to the glycosidic bond. Okay. What about protein? Yeah.
So they give starch or maltose, sir.
Okay.
Sir, no, sir. I'm asking, sir. Doubt, sir. Is it starch or they give maltose, sir, when 2 glucose link up, sir?
No. So what I am asking, I'm not. So what? Once again, pardon.
Sir, like when 2 glucose molecules link up, sir.
Okay.
What do they give, sir? The end sugar they form, sir. What is
Yes.
that, sir?
Suppose a monosaccharide is there. Imagine. Okay. Glucose is a monosaccharide, isn't it? Suppose another glucose molecule is also there. Okay. Suppose these two are united together, form as a disaccharide, isn't it? Sucrose is a disaccharide, and also maltose is a disaccharide, and also lactose is a disaccharide in our body, isn't it? Each sugar molecule, they are having 6 carbon atom. They united together with the help of the bond is called as glycosidic bond. As a result, you will get it 2 monosaccharide united together form as a disaccharide. More than one sugar molecule united together the glycosidic linkage, as the result of formation of what is that? Polysaccharide. One glucose molecule unite with another one molecule with the help of the glycosidic. One amino acids connected with another amino acids with the help of the bond is called as peptide bond. Okay, suppose one glucose with one amino acids connected with the bond is called as glycopeptide bond. Okay, glucose as well as the peptide. I mean glucose molecule as well as the amino group united together, that is called as glycopeptide. Okay, you got it, my dear students. Ajay, any doubt in this portion? Are you clear now?
Yes, sir.
Okay, go ahead. So go to the next one. This is the normal HPA peptide bond is there. HPA yes peptide bond is also. See the diagram. See the here. Just see the position. See the first amino acid. Which one is there? Valine is there. Here is also valine. What are the difference between these two? See here. And second one is histidine. Second is also histidine. Third one is leucine. Leucine is also present. The fourth one is 309, isn't it? And fifth one is, what is that? Proline. What about the sixth one? Just listen here. Glutamic acid is here. Here the valine is there. Don't forget, my dear students. Sixth position of the amino acids will be changed. Okay. In the HPA and HPS. What is the difference between the sixth position only changed? Okay. Glucose and glutamic acid here. Here the valine is there. The HPS containing valine, but at the same time HPA is containing glutamic acid. Don't forget, my dear student. These are the difference between these two peptide bonds. Peptides, sorry. Okay. See that. The defect is caused by the substitution of glutamic acid by valine at the sixth position of the beta globulin chain of the hemoglobin molecules. Once again, repeat for you. Here already I asked the question. Okay, the Ajay also answered it. Okay, other students, please listen carefully. Okay, what is the defect? Okay, what happened? Sickle cell anemia, how it will be happen? You know about the protein molecules. There are 2 protein molecules have been reported. One is called as, what is that? Yes, another one is A. Okay, where is the position? The sixth position is replaced by, okay, glutamic acid replaced by which one? Valine in the sixth position of the beta globulin. That is very, very important. Here the 3 question is that glutamic acid replaced by valine. First one, which position? Sixth position. How many amino acids are there? 7 amino acids are there. Which globulin molecule chain? Which chain? Which globulin chain? Beta globulin chain. So hemoglobin contain how many chain is there? Will you please, anybody? How many chains? How many globulin molecule present in? Anybody else, my dear? Ajay, Dhanalakshmi, Varsha?
Sir, don't know, sir.
You don't know why? Already we discussed, my dear. Already we discussed. What about Dhanalakshmi?
No idea, sir.
What about Varsha?
So what is the question?
Each and every hemoglobin, how many globulin molecule present in it?
So 4.
4. Very good. Will you please tell me how many? How they are differentiated, divided? Each and every hemoglobin molecule containing 4 globulin chains, right? 2 alpha chain, 2 beta chain. Don't forget, my dear. This is very, very important. 2000. Before, I think the question is asked. Each and every hemoglobin molecule containing, what is that? 2 types of chain is there. One is called as alpha chain, another one is called as beta chain. 2 alpha chain, 2 beta chain is also there. So they carry, what is that? 4 oxygen molecule. Don't forget. Very good, Ritik. Varsha. Good. Others also, please. You are answering, you are trying the answers, but at the same time, be confident. Okay. The substitution of the amino acid in the globulin protein result due to the single base substitution at the 6th codon. Note it down, my dear students. Which codon is replaced? Sometimes they may ask the question. GAG is replaced by GUG. See the position. See here. GUG is replaced valine. So GAG is also glutamic acid. So the GAG is denoted as a glutamic acid. The glutamic acid is replaced by which one, GUG, that is denoted as valine. Sometimes they may ask the question. GAG, glutamic acid. GAG, guanine, adenine, guanine, that is glutamic acid. GG, note it down, my dear. Here valine is there. So near which one, U is the answer, UV. So U is center, I will get it a valine. Don't forget, my dear. GUG. It is a triplet codon denoted as a valine. So center is, there is a U is there. Here the valine is indicator. Otherwise called as valine is the amino acids, just like nearest one. Here, see that GAG, G. A is there. Okay. See that one? Glutamic acid. Don't forget. GAG, glutamic acid. GUG is a valine. Okay. It can be replaced by the 6th position of the peptide chain. So how many globulin molecules present in the hemoglobin? 4 chain is there. 2 alpha chain, 2 beta chain is also there. Then which place they replaced? 6th position is replaced by the beta chain only. It's not alpha chain. Don't forget, my dear. The substitution will be which one? GAG, GAG. It's replaced by GUG. This is valine. This is glutamic acid. GA is also called as glutamic acid, like that you can imagine. Okay, at the time of examination point of view. For the next one, see that the same thing is also replaced here. See the 6th place. Okay, replaced by the valine. See here the glutamic acid is there. So replaced by the valine molecule in the 6th place. That's why they have put a arrow. Okay, so this is HPS. Don't forget. See that. The mutant hemoglobin molecules undergoes the polymerization under the low oxygen tensions, causing the change in the shape of the RBC. Suppose it will not carry these changes due to these changes. Which one, the nucleated changes, what happened, the protein molecule shape also changed. What about the structure of protein molecule? How many structures also studied in protein molecule? According to the structure, protein molecules are divided into 4 types, isn't it? What are they? Do you know? Do you know, my dear students? Primary structure, secondary structure, tertiary structure, and quaternary structure. Okay, the hemoglobin also comes under the quaternary structures. That's why they have, what is that? 4 chain, 2 alpha, 2 beta. Okay, so each hemoglobin molecules, they are having 2 alpha chain, 2 beta chain. At the same time, they are carrying, what is that? 4 oxygen molecule. Okay, see here. The mutant, as a result of the changes of CAGs and also CAG, what happened? There is a change is there. As a result, the oxygen tensing causes the changes in the shape of the RBC from biconcave disc to elongated sickle-like structure. As a result, it will not carry the proper oxygen molecule from one place to lungs to, or whatever it may be, from the respective places or target. So that condition is called as, what is that? Sickle cell anemia. It is also one of the autosomal recessive single gene, pair of gene, single pair of gene. Don't forget. Okay. So remaining things is that, okay, this one, phenylketonuria. It is also another one, but in this category, this is inborn error of metabolism. Sometimes they may ask the questions, which one of the following is the inborn error metabolism? Phenylketonuria. Don't forget, my dear. It's also inherited as an autosomal recessive type. All are recessive character only. Already we discussed it. Autosomal recessive only, not dominant. 2 only dominant is there. Already we studied skin color. Okay, when we were studied, which one, chromosomal. That time we already studied that things also. Here, phenylketonuria is also inborn error metabolisms and also called as autosomal recessive type. Due to the recessive gene only, it is, what is that? The disease is caused. what happened here? The affected individuals lack an enzyme that convert that amino acids phenylalanine into tyrosine. Don't forget, my dear. What happened? No, the particular recessive gene is absent. What happened? The particular enzyme, it will not produced. The particular enzyme is not produced. What happened? You know, the conversion of amino acids. Which one is phenylalanine? Is the aromatic amino acids converted into tyrosine. These are all are, what is it? Aromatic amino acids is that. Aromatic means you know the structure, hexagonal structure, and center there is a ring structure is that phenylalanine. The conversion is not possible. As a result, what happened? As a result of this phenylalanine is accumulated. This conversion is not possible. What happened? The phenylalanine accumulated and converted into phenyl pyruvic acid and other derivatives. This is very, very important. Due to this one, what happened? The enzyme is not involved. Conversion takes place. Lack of the particular gene. The enzyme production is also lack. As a result, the amino acids conversion is also lack. So as a result, what happened? The phenylalanine conversion into tyrosine. The conversion also same. So many times they were asked. Okay. So as a result, phenylalanine accumulated in the body. The phenylalanine accumulated. What happened? No, the phenylalanine converted into phenyl pyruvic acid. Not only the phenyl pyruvic acid, and also converted to some other form, some other derivatives also. Okay, see that one. So dietary protein, the phenylketonuria, how it will be comes out. All are comes out the nitrogen sources. Which one is the nitrogen sources? The protein is the nitrogen sources. Whether it may be dietary protein, sometimes it may be tissue protein, because after the digestion of protein, it's stored in the tissues. Okay, these protein molecule converted into phenylalanine. It's nothing but the amino acids, isn't it? The phenylalanine converted into tyrosine with the help of the enzyme is called as phenylalanine hydroxylase. Don't forget, my dear. This is very, very important. Which enzyme is lack here? Phenylalanine hydroxylase. Hydroxylase means addition of hydrogen, OH. So when you are adding the hydroxyl ion, the phenylalanine converted into tyrosine. Okay. So this reaction is suffered. What happened? No, the phenylalanine deposited, the phenylalanine converted into the, it also entered into the other pathway, phenyl pyruvate. The phenyl pyruvate is also further divided in phenyl lactate and phenyl tactate, like that. So this is the oxidation of protein synthesis, catecholamine, tyrosine, and melanin. The tyrosine responsible for the, these are the process. Oxidation of protein synthesis, catecholamine, and thyroxine and melanin. The melanin is also one of the pigment. So this reaction is suffered by the lack of this enzyme. What happened? This process completely lack. So it will lead to some disease, disease called as what is that? Phenylketonuria. It can be excreted in the urine. Okay. So there is a reason. Okay. Up to this level, we will finish it. To remaining things we will see and the next classes, my dear students. Okay. Don't forget, my dear. So what are the things already we discussed here? You know, there is a 4 diseases already we discussed. One is called as hemophilia, and second one is hemophilia. How many types is that? A and B and C is also there. And cystic fibrosis. What is by cystic fibrosis? The mucous membrane, which enzyme, which protein, which chromosome is that? That things also already we discussed. And sickle cell anemia. What are the pairs also there? There are 2 genes also responsible. How it will be? What are the chain? What are the changes which occurs? Which position is changed here? First one, 7th one position, which 7 chromosome, you just note it down. Here 6th position will be replaced the amino acids. That is also very, very important. Okay, my dear students. So don't forget. So, and the last one is already we studied. Which one, you know? That is called as what is that? Phenylketonuria. Phenylketonuria is a very, very important. It is an inborn error metabolism and also recessive triad. It produces the lack of an enzyme. That enzyme is called as phenylalanine hydroxylase. The conversion of the phenylalanine into tyrosine. Don't forget. Okay. So we'll see the remaining things on the next classes. Thank you for the participation and thank you so much. Okay. Thank you so much, my dear.
Thank you, sir.
Thank you, Bob. Thank you, ma'am.
Thank you, sir.
Thank you, ma'am. Thank you.